Hi, my name is Ratner and I am a senior in the Tulane School of Science and Engineering. I am studying psychology and cognitive science, and I have been working in Dr. Galazo's Cellular Neuroscience lab since my sophomore year. I study the brain circuitry involved in motor movements, and specifically we are looking at the contribution of certain types of neurons in voluntary motor movement. All right, let's take a look around my lab. So part of what we do is stains of brain tissue for different markers in brain tissue so we can highlight different areas and hone in on what's going on. My research is primarily seeing if there is another type of neuron that projects from the brain to the spinal cord that would create those motor movements, whether they are voluntary or involuntary, like neurodegenerative diseases. So this would be ALS, amyotrophic lateral sclerosis, or MS, multiple sclerosis, Huntington's disease, Parkinson's. So let's go take a look at some neurons. Over here, we are looking at a big old clump of neurons. We have reconstructed where these neurons project from. And so with this, we can see what areas of the brains that these neurons, while their bodies are in one area, their projections lead to another. It's like hearing about a cancer with no cure. And with neurodegenerative diseases, so much of the population falls victim to them at a certain age. The research that we are doing, its implications can greatly help that population. So over here, we have some pipette beveling, and we bevel to create an opening on one side. That way, when we perform injections, the liquid can flow through. The device is a microscope that is connected to a platform with a tool that propels in a circle. This also has a bit of a stone to help be a bit more abrasive to create that opening. We have a bit of a lever that we screw up and down to make sure that the capillary tube does not have too much pressure on it. It would snap. It's very delicate. That was a quick tour of our lab, and thank you so much to Belle for some help with some demonstration. I spent the early days of my time in this lab carrying around my lab notebook and just jotting down as much as I could. The first few pages are me learning how to extract DNA and run gels, which are things that I do almost every day in the lab. Scientists are innately curious people who get excited about solving a problem and finding solutions. It makes science exciting.